Assalamualaikum and a very good afternoon to all of you. Welcome to INBIOSIS webinar lecture series number 10 in year 2022. I am Dr. Nurkhalida, your moderator for today's webinar. So the video that we played just now captures the gist background of our institute, INBIOSIS, located in University Kebangsaan Malaysia in Bangi, Selangor. I hope it has given you sufficient insight on the institute as a whole. So, for those who are interested to collaborate with us, you are most welcome to do so. Feel free to browse INBIOSIS website and social media for the latest updates and info. I feel so grateful today that all of us are granted good health and time to attend our webinar. So far, this is our 10th webinar for this year. There will be many more webinars to come, and I hope all of you will join us again in the future. Our honorable speaker for today is our own research fellow, Dr. Kamalrul Aslan Azizan. Before we start, allow me to share with you Dr. Kamalrul's CV. Kamalrul Aslan Azizan completed his PhD degree from UKM in 2014. Specializing in the area of microbial metabolomics. He is currently working as a research fellow or senior lecturer at the Institute of System Biology or INBIOSIS in UKM. His research focuses on the application of metabolomics to study plant and microbial metabolomics. He has vast experiences in GCMS-based metabolomics, LCMS and chemometrics analysis. At present, he is using metabolomics approach to understand allelopathic interactions. Without further ado, let us welcome Dr. Kamal Aslan to share his experience with us. The screen is yours, Doctor.
Okay, thank you, Dr. Khalida. Can you hear me? Is my voice clear? Hello? Okay, right. Okay. First of all, thank you, Dr. Khalida, for the kind introduction. And also I'd like to thank INBIOSIS for giving me the opportunity to speak about my research in this webinar. So without further ado, let me share my slide. Can you see my slide?
Yes, Dr. Kamal. We can see your slide.
Okay. Right. So for today's webinar, so I'll be talking about metabolomics. So the title of my presentation is Metabolomics for Biomarker Discovery, Key Signatory Metabolic Profiles for the Identification and Discriminations of Plants and Microorganisms. Right. So before we further go into my talks, so first I would like to do some introductions about what is metabolomics. I believe that everybody here is familiar with metabolomics. For those who are not familiar or first time hearing what is metabolomics, so basically metabolomics is the study of identifications and quantifications of small molecules or what you call as metabolite that present in biological systems. Okay, so metabolomics is heavily depend on the high throughput analytical chemistry and material analysis, whereby these two important tools are very crucial in metabolomic studies for us to do data analysis so that we can actually capture the relevant biological information that we need from these so-called metabolites. Right. So in metabolomics, we encounter with what is called as metabolic profiling. So basically, metabolic profiling is a way of is a tool that we can use to actually do quantifications or measurements of metabolites, which involves excretions, separation, and also analysis. Now, in my title, I mentioned about biomarkers. So what is biomarkers? So basically, biomarkers is a biological characteristic that is objectively measured and evaluated as an indicator of normal biological or pathological process. So basically, this biomarker is very important so that we can actually use this as an indicator so that we can actually explain what is happening inside the biological system, whereby, for example, like if a biological system is challenged by specific environmental condition, then we can use this biomarker to actually explain what is going on inside the biological system. Right. So when we talk about metabolomics, so in this case, I'm be focusing on plant and microorganism. So as all of you know, plants are part of a rich ecosystem here in our surrounding, including numerous and diverse microorganisms that also coexisted with plants. So when we talk about plant, so in terms of human nutrition and also crop improvement, plants play an important role in our life. Plant as a source of our food and also source of our economy. So in this case, when we talk about plant, of course, we'll be talking about planting or cultivations, environmental stress response that is being faced by the agriculture sector, for example. What are the mode of actions, how these plants and microorganisms, including act or respond towards certain conditions, source and traceability, or when we talk about adulteration, for example, like food security, for example, harvesting and also quality control assessment, we want to have a good quality products towards the end of, for example, agriculture or towards co-cultivations of micro. So how metabolomics can tackle this issue? Of course, when we talk about metabolomics, we cannot run away from metabolites because we use metabolite. We use metabolite to investigate the biological system, how it reacts towards environmental stimuli. And of course, we need to monitor and assess this metabolite using a systematic approach. In this case, we have untargeted or targeted approach using mass spectrometry method, for example, for us to monitor and assess the metabolite. And of course, at the end, we can use this information for optimization strategy for quality improvement. Now, why metabolomics? So, of course, when we talk about metabolomics analysis, what we want to do is not just profiling, getting what are the chemicals or metabolite inside this biological system. We want to do a comparative metabolomics, whereby we want to compare between two conditions, for example, so that we can actually capture these so-called biomarkers that we can use it to improve the quality of the biological system that we are studying. So from there, we can actually use metabolomics to identify and discriminate samples or subjects accordingly, right? So of course, in metabolomics, we need to have a very good or at least an efficient sample preparation, because without good sample preparation, we cannot capture the metabolites that are very important in this biological system. So the idea here is for us to try to maximize the metabolic profile that we can obtain using this sample preparation. Of course, by using appropriate analytical instruments such as mass spectrometer coupled to liquid chromatogram or gas chromatogram, we can actually increase the sensitivity of this metabolic profile that we want to capture. And of course, metabolomics depend heavily on chemometrics analysis to identify these key biomarkers. So when we talk about chemometrics, of course, we need to employ univariate and also multivariate analysis, which we be explaining later in this talk. Now, so here, the most important subject, there are two subjects that are going to talk in this webinar. So the first one is actually plant metabolomics. So what are the targets that we want to, when we do plant metabolomics? Of course, we want to capture the metabolites, but what metabolites? Of course, we want to capture both primary and also secondary metabolite. So of course, when we talk about primary metabolite, it's all about the core metabolites that are very important for the development or the growth of the plant itself. Whereby secondary metabolite is more towards defense mechanism of the plants toward environmental stimuli or infection against pathogen and etc. So how about the sample? So for plant metabolomics, usually the samples we can obtain, we can use either leaf, or bark, flower, root, or even the whole plant. And when we talk about methods for plant metabolomics, the most common methods that we usually use is liquid extractions using biphasic extraction, employment of two different solvents with different polarity, or maybe we can use solid phase extraction or SPE, or maybe SME, solid phase microacidations. And the aim or the objective of plant metabolomics usually is to do, of course, comparative to identify key physiological process that is happening to the plants, whereby we can actually further understand what is going on inside this plant. Now, as for microbial metabolomics, it's actually similar to those in plant metabolomics. So basically, of course, we want to capture primary and also secondary metabolites. Of course, when we talk about primary, again, primary metabolites such as amino acid or organic acids, these are very important metabolites that we can actually alter so that we can do some optimizations, in this case for microbial metabolomics. And of course, secondary metabolites is very crucial for us humans to use, especially in the medicines sectors and in this pharmaceutical sectors and etc. Now, when we talk about sample for microbial metabolomics, it's a bit different from plant metabolomics. So here we have intracellular and extracellular. So basically when we culture our microbes, so the brood, we get the brood. So the brood inside is called as an extracellular, while the intracellular inside is actually what are the metabolite inside the cells. So there is the difference between intra and extra. Also, we can do mono or co-culture, and then we can try actually to separate this intra and extracellular and actually capture the metabolites that we can obtain from this intra and also extracellular. As for the method, again, is a bit different from plant metabolomics. Yes, we can employ liquid extraction, we can employ SPE, SPME, etc. However, there is another crucial step for microbial metabolomics is actually the quenching part, whereby we need to actually employ this quenching technique so that we can actually separate the intra and the extracellular. And again, the aim or objective for microbial metabolomics is actually similar to plant metabolomics. Of course, we want to do comparative at the end. Again, comparative metabolomics is very important when we're designing our experiment involving metabolomics. Of course, we want to know the biological activity, the growth condition, and also, of course, to identify the key physiological process that is happening in our microbial system. Now, so moving on to the metabolic profiling workflow. So this is actually a schematic representative of how a metabolic profiling workflow is carried out. So first, we have our sample preparation. Of course, we have our biological sample, we have technical, and of course, we need to have a replicate because this is very important because size sample is matter in metabolomics. You may have to increase your size sample in order for you to get reliable information. In this case, you want to get good data involving your metabolomics data. So here you want to have, because you are going to use either peak area or peak height or maybe intensity of your data. So again, size sample will actually affect the way you process your data because here will determine whether you need to do either parametric or non-parametric method for your univariate analysis, right? So moving on to the next step is the sample analysis and data acquisition. Of course, when we talk about sample analysis and data acquisition, we cannot run away from mass spectrometry methods because mass spectrometry method is the core for metabolomics. So whereby we use or depend heavily on this technique for us to not only capture the metabolite, but also to identify the metabolites. So currently, there are numbers of analytical instrument that you can use to do metabolomics. So the common one is, of course, liquid chromatogram and gas chromatography coupled to mass spectrometer. You can also do metabolic profiling using HPLC, whereby you need to have a standards because you cannot do identification using HPLC. And there are a lot of publications that actually use NMR for metabolic profiling, especially in the area of metabonomics, whereby they utilize or they analyze fluid sample from body, from urine, from blood, and etc. So the third step is the data processing. So it's very crucial step in metabolomics, because here you need to do data alignment, you need to do filtering, deconvolution, normalization, and data mining. So here is where you obtain your data metric that consists your samples, your numerical data, either peak area, peak height, or even intensity. And of course, nowadays, you have a numbers of web-based tool or even commercially available software that you can use to do data processing. And the last part is actually the data analysis, whereby you need to do some normality tests, univariate analysis. Of course, you need to do chemometric method in comprise the unsupervised and also supervised metabolite. This is very important because here you want to do classifications of your sample. Do classification of your sample. Of course, you want to find the biomarkers that you can do, that you can use to explain what is actually happening in this sample or biological system that you want to study. And of course, you want to do some biological interpretation using pathway arrangements, SEA, and etc. Right, so we're going to go a bit deeper on plant metabolomics. So here is the schematic diagram schematic representative diagram that show how plant metabolomics is being carried out. So again, you have your design of experiments, and then you have your sample acquisition, you have sample preparations, and then you subjected this sample to machine to analytical instrument to do further sample analysis, and then followed by data processing. And of course, from there, under data processing, you have a numbers of steps that you need to do so that you will get the so-called data metric. And then from there, using this data metric, you use univariate and also multivariate, or basically chemometric analysis to get classification, biomarkers, and biological pathway. So in this process, so basically these are the process of plant metabolomics. So of course, again, data processing is very important in metabolomics. And of course, for classifications, biomarkers, and certain biological pathway analysis also depend on material analysis, such as PCA and PSTA. Right. As for microbial metabolomics, again, is quite similar to plant metabolomics, but there are a few steps that is quite different. So again, earlier what I mentioned about quenching. So the idea of quenching is because most of the metabolites in microbial metabolomics, especially in the intracellular, have a high turnover rate. And then you have problems such as intracellular leakage, cell degradation, and poor extraction reproducibility. So in order to overcome this limitation, so we employ quenching solutions. So when we talk about quenching solution, there are numbers of quenching solution that we can actually employ. Using, for example, glycerol saline, using hot ethanol, and etc. And each of these actually, you need to actually have a basic understanding on what is going on with your microbe. For example, like if you have a gram-positive systems, you cannot simply use ethanols or harsh solvent to do quenching. So you need to have a solution that is actually well suited with the microbial system that you're going to use. Right. So that's all for introduction of metabolomics. Now going to move on the works that on the research that I've actually carried out. So for this talk, I'll be using example of metabolomic analysis to differentiate Lactococcus lattice. Then I'll also be talking about metabolomics approach to determine the phytotoxicity activity of Wedelia chirobata. And finally, some collaborative projects that have been involved for the past year. Right. So first, I'm going to actually explain on the metabolomic analysis to differentiate Lactococcus lattice. So basically, this was my PhD project. So basically, the idea is to actually to understand this Lactococcus lattice when growth at different growth conditions. So Lactococcus lattice is actually a member of mesophilic group of bacteria. One of the main constituent in both industrial and artisanal starter culture. So basically, the Lactococcus lattice has been regarded as grass status, generally regarded as safe, whereby this strain is heavily used to produce cheese. So the idea of this work was to do a metabolic profiling under different culture conditions. Right. So initially, what we did was we tried to actually capture the metabolite or obtain the metabolite profile from this Lactococcus lattice when in response to different growth conditions. So we employ conditions of 30 degree and 37 and a condition at agitation which is at 150 RPM. So from here, of course, we did quenching. So we focus on the intra and also extracellular analysis, whereby we separate it using glycerol saline solution to extract the intracellular and also the extracellular. And from there, we come up with this so-called heat map and also some biological pathway. So basically here in this study, because we are actually doing a microbial metabolomics, of course, we cannot run away from capturing this so-called primary metabolites or core metabolite. In this case, we capture a lot of amino acids, which is actually the pivotal metabolites for this Lactococcus to grow. So basically here is the heat map that showing metabolite changes, especially on the amino acids when cultured at different growth conditions. Right. So from there, a work by Shuhaila, which was supervised by Associate Professor Dr. Syaratul Al-Kain. So here we actually further the study by comparing the protogenic amino acids in Lactococcus when grows in different agitations. So here we employ numbers of different agitation speed and try to see what are the difference in terms of the progenitor amino acids, how these metabolic changes occur in this Lactococcus in response to different agitation. So from here, we found there are huge differences, especially for the amino acids, and we managed to actually, managed to actually separate these metabolites based on these agitations. And we found out that Lactococcus grows at 150 RPM, show strong metabolic changes, especially in the protogenic amino acids. Right. So using these informations, we then move to Fluxon Study. So basically we use a 13C label carbon source. We feed it to this Lactococcus like this, and then we try to infer into the central carbon metabolisms. So again, we use protogenic amino acid as our indicator to see what is actually going on inside the central carbon metabolism. So from here, we use different label 13C, different ratio of 13C. We feed it to the Lactococcus, and then we use GCMS. From there, we quantify this label, and we come up with this pathway whereby we can see huge differences, especially for Lactococcus in response to agitation condition. So from here, the CCM itself, the metabolites, so especially the protogenic amino acids, actually provide a good metabolite discrimination. And here, these informations can be actually used for further metabolic engineering of this strain. So based on the finding that we have obtained, we can conclude that metabolic profiling of Lactococcus lactis shows significant metabolic changes in response to different growth conditions. Then it's revealed that partial least square discriminant analysis of PSDA revealed that changes in of amino acid and fermentation products influence the separation thread, especially for the agitation conditions. And branched and aromatic amino acids, alanines, aspartate, glutamate, and glycine were identified as the common amino acid observed in Lactococcus lactis and can be regarded important for reliable carbon TC, carbon 13, this metabolic flux analysis for Lactococcus lactis. Right. So that's all for microbial metabolomics. Now we're going to move to metabolomics approach to determine the phytotoxicity activity of Wiederia tuberata. So basically we are going to move into plant metabolomics. So here in this research, actually, we are focusing on this plant called Wiederia tuberata. So Wiederia tuberata is actually also locally known as Serundella or creeping daisy. It's actually a member of family Asteraceae. So it's a mat for forming perennial herbs with a creeping climbing habit. So this plant is actually widely cultivated as ornamental or ground cover plant in public landscape such as public parks and etc. And this plant is actually attractive, glossy, have a dark green, bright yellow, daisy-like bloom flower. So this plant has a very wide ecology tolerant range. So basically, it's spread rapidly and is difficult to eradicate. Also, this plant has exhibit significant phytotoxic activity and also allelopathy interaction, whereby it can actually inhibit other plants through chemical that is secreted out surrounding this plant. So there are actually 6 Wiederia species in the literature that have been reported, but the main Wiederia species that have been mostly reported is Wiederia chinesis, whereby this species has actually been used as a tea to be consumed as a tea. Also, there are numbers of publications regarding Wiederia tuberata and also Wiederia biflora. So in this research, we are focusing on the Wiederia tuberata as we found out that this Wiederia tuberata has better or significant phytotoxicity activity compared to other species. Right. So in this study, what we did was we focused on, of course, on the phytotoxic activity. We tried to get the global metabolic profile or metabolic profiling of this plant. And then, of course, we also focus on the soil where this plant grows. So basically, we also did a so-called soil metabolomics. And then we compare it with the metabolite that we obtain from this plant. And of course, we also focus on essential oil. So basically, we extract the essential oil from this plant and also study their phytotoxic activity. Now, so the idea of this study is actually to look into the metabolic profile, especially compounds that have this phytotoxicity, where it can actually be used as a herbicide to inhibit other, for example, growth of wheat and etc. So from here, we try to actually correlate this metabolite with environmental factors. So in this case, environmental factors, of course, we did a study on the soil and tried to correlate this information with the metabolic profiles of this plant. So the main objective of this study was what are the metabolites that contribute to this phytotoxic activity, and then what are the factors that contribute to these activities, and what are the mode of action or mechanism for this activity. Now, so initially, we started with getting the right solvents to capture the right metabolite. In this case, the metabolite that contribute to the phytotoxic activity of this plant. So in this study, we use ethanol and water to extract this metabolite. Of course, we also employ different solvents, biphasic solvents, and using metabolomics and of course using multivariate analysis, we compare which solvent yield the best metabolite with strong phytotoxicity activity. From here, we found out that ethanol extract gave the most significant phytotoxicity compared to other solvents. So from there, we moved to LCMS. So we actually employ GCMS to do the comparison, and from there we moved to LCMS. We want to further do the metabolic profiling. Of course, we talk about GCMS. What we managed to capture are mostly primary metabolites. Yes, we also do capture secondary metabolites, but LCMS give a better, or in these terms, in this case, more sensitive analysis, especially governing the secondary metabolite that we want to capture. So using the information that we obtain from the GCMS, we actually use LCMS to further analyze our ethanolic extract. So from there, we found a number of secondary metabolites that actually can be a marker to actually identify this Weigela triobata. So from there, from the metabolite profiling that we obtain, we actually captures a number of metabolites such as caffeine acid derivative and also D13 carotenoic acid or Ka, which can be served as a phytochemical markers or index for quality control and also identification of this Weigela species, especially Weigela triobata. Now, from there, we move on to the metabolomics analysis of these leaf, litters, and soil. So, in this study, we compare the metabolite profile from the leaf litter, and also from the soil where this plant grows. So, again, in this study, we use LCMS to capture both primary and also secondary metabolites. So from here, we also actually did biological bioassay studies. We tried to actually compare what are the phytotoxic activity when compared between this leaf, litter, and soil. What we found was, of course, increasing of ethanol extract concentration of all extracts significantly inhibited. In this case, we we test this extract on lettuce to actually establish what are the phytotoxicity, what are the phytotoxic activity percentage that we can obtain from this extract. So from there, we found out that actually soil accepted the most significant phytotoxic activity. However, in terms of the metabolite that we could capture, the soil end up gave a very little metabolites. So here, what we can postulate was the sample preparation, especially for soil metabolomics, is quite difficult compared to leaf extraction because the matrix itself is much more complex compared to leaf. So in that case, we actually go deeper. We actually proceed with the analysis of soil metabolite only. So in this study, the dynamic linkage between phytotoxicity and metabolite in Weigela turbata soil. So basically here, we focus solely on this soil that we obtain under this Weigela turbata. So here we did actually a time series. So we collect soil for 11 months, and then we subjected this soil to metabolic profiling. So again, we redesign our experiments because we want to maximize the metabolic coverage that we should capture compared to the previous studies whereby we only have a very little metabolic profile from this soil metric. So in these studies, we actually redesign our experiments, and then we also did the time series. So we try to compare the metabolic profile for 11 months. So we collect the sample with 1 month interval, and then we did extractions and we compare the metabolic profile. Of course, we also did the phytotoxic activity again using this sample and we try to compare, and we compare the phytotoxicity, which month gave the highest phytotoxicity. And from there, we try to actually correlate this phytotoxicity activity, the metabolites, with the physical-chemical properties of the soil. In this case, we actually try to understand how the soil physical and chemical properties can be correlated with this phytotoxic activity and also the metabolite. So here we did some studies, some characterizations on the micronutrient and also the on the micro and micronutrients of this soil. So here we actually measure the calcium, the magnesium, the kalium, basically the micro and also the micronutrient of the soil that we capture, that we obtain. And from there, we found that, of course, the result revealed strong variability in soil properties and obvious temporal variations in the soil phytotoxicity over the 11 months period. And among the soil properties, we found that the SOM, or the soil organic matters, the EC, the total nitrogen, and soil nutrient present the most contributions in differentiating soil according to the time and sapling and the strength of phytotoxicity. Of course, these factors also influence not only the phytotoxicity but also the metabolites that contain in this soil. So from there, we also actually did a study on the essential oil of this plant. So basically, in this study, we did a comparative metabolomics again by comparing metabolites of this Weigela turbata grown in different conditions. So in this case, we compare plant that we actually cultivate in the field condition and also in the growth chamber. So from there, we extracted the essential oil, and then we compare the metabolite profiling using GCMS. So here you have the FTIR analysis of the essential oil itself. We found quite interesting peaks or regions when we compare with other commercially available essential oil, such as cinnamon acid, eucalyptus, and etc. So so here again, we try to compare what are the metabolites that change when we cultivate this plant in different conditions. So from here, we found out that when plant grows in the control conditions, the 6-terpene hydrocarbons increase, whereas the essential oil that we obtain from plant that were grown in field conditions have a large amount of monoterpene. So from here we can see there are strong variation of metabolites. From there, again we did a phytotoxic activity of this essential oil, and we found that essential oil that were grown in growth chamber, essential oil obtained from growth chamber have a better phytotoxicity compared to the field, and were contributed by this monoterpene. So here we employed this Chemometrics Method, whereby we combine this metabolite data with the biological data activity, in this case the phytotoxicity, and from there we actually pinpoint the biomarkers or the metabolite that are responsible for this phytotoxicity using PLS regression method. So again, here you can see that the idea here is the PLCA regression revealed that the seed germinations of BD rice and lettuce were susceptible to 𝐕𝐸 turbata essential oil grown in the controlled conditions, and nano may contribute to the phytotoxicity of this EO. Right. So from there, we actually further expand our studies on how this essential oil actually change or disrupt the growth of this 𝐕𝐸 rice and also lettuce. So basically, using the essential oil from the growth that we obtain from the control conditions, so we expose this essential oil to 𝐕𝐸 rice and also lettuce seed. And from there, we did a metabolomic analysis on the 𝐕𝐸 rice and also the lettuce that's been exposed with this essential oil. So from there, we use different different concentration of essential oil, and then we capture the metabolic profiling, and then we use multivariate analysis to do a comparative analysis of the metabolite that had been altered due to the essential oil. So here we have this heat map that actually tell us how this essential oil alter the metabolite compositions in this 𝐕𝐸 rice and also in the lettuce. If you can see here, different concentration of essential oil actually give different metabolite alterations. So from here, we found that this essential oil from this 𝐕𝐸 turbata change the fatty acid bias disease and also affect the elongations of this plant. So this result indicate that 𝐕𝐸 turbata essential oil can cause alterations in this fatty acid composition and lead to the inhibition of root growth of 𝐕𝐸 rice and also lettuce. Now, beside that, we also did an isolations of soil microbial from the soil that we obtained under this 𝐕𝐸 turbata. So this work is actually still in progress. So basically, we try to actually understand what is the role of soil microbes in mediating the soil phytotoxic effect in the 𝐕𝐸 turbata. So previously, we did an experiment regarding the soil metabolomics. We did actually in that study, we also compare between non-sterile and also sterile soil, and we found out that in non-sterile soil give better phytotoxicity compared to sterile soil. So basically here, we postulate that this microbe community, this soil microbial community, may give do not give a great influence on the phytotoxicity itself. However, there are several studies that indicate that invasive species like 𝐕𝐺 turbata can actually alter the soil microbial community inside the soil, inside this soil. So therefore, we actually did some isolation, and we found out we obtained a numbers of isolate. So we actually use biolog microplate to further characterize this isolate that we obtained. And interestingly, for this work, we actually can correlate back with the metabolic profile from the soil that we obtained. Especially, we found this particular compound, a Cider-4 compound, which actually helps this 𝐕𝐺 turbata to get more iron from soil. So from that hypothesis, from that result, we actually try to isolate microbe that have effect on the phytotoxicity and also the growth of this 𝐕𝐺 turbata plant. So as a conclusion for this study, so we found out that 𝐕𝐺 turbata is a noxious invasive weed containing diverse activity compound with a broad spectrum of biological activities, especially the phytotoxicity activity. Here we found that exposure of 𝐕𝐺 turbata extract can cause alteration in the fatty acid composition and lead to inhibition activity, especially on the essential oil. And combinations of metabolomics and biological data or chemometrics can provide a metabolic understanding and bioprospecting potential for 𝐕𝐺 turbata. Right. So as for now, I also involved in a numbers of collaborative project. This is actually a project with FRIM. So in this study, we did an untargeted metabolomic approach using 𝐈𝐍𝐁𝐈𝐒 to actually classify tonkat ahli samples and to determine metabolite variations in tonkat ahli roots that have different age, 1, 4, and 11 years old. So again, from here, we also try to actually do an integration analysis whereby we have the transcriptome data, and then we try to actually integrate this data, and we found interesting pathway that related to the bioactive compounds, especially on the 𝐒𝐞𝐜𝐿ectone. Then I also involved with the work on the essential oil of agar wood. So basically here we try to actually do a comparative metabolomics of healthy agar wood compared with naturally induced agar wood and synthetically induced agar wood. So from there we did a comparative metabolomics of the essential oil, and then we try to compare the metabolite and find out if the synthetically induced essential oil give better quality compared to naturally induced essential oil of agar wood. I also involved with the study on the phytotoxic effects and identification of potential allelochemicals from Tunera subulata. So from here, this work was carried out with UNIZA. So from in this work, actually we have two major species of this Tunera subulata. So we did a metabolic profile of the leaf extracts and then compare the metabolites. And from there, UNIZA actually did the integrations with the phytotoxic data and come up with the explanation on how this plant exhibit the so-called phytotoxic activity. So as a take-home message, so again, metabolomics allow metabolic profiling to elucidate metabolite and related metabolic pathway. So of course, metabolomics is a powerful tool that enables the comprehensive study of plant and organism. So I have actually shown you two examples how we can use metabolomics to understand the conditions of Lactobacillus lattice for microbe and also Vigilante rota for plant. And finally, metabolomics have contributed significantly towards an understanding of plant and microbial science and identification of key pathway. Of course, at the end, we need to do integrations with other omics via system biology to further understand this biological system that we are studying. So before I end my talk, so a little bit on Central of Omics Data Analysis or CODA. So basically, INBIOSIS through CODA is a one-stop solution for omics data analysis. So here we offer data analysis for transcriptome, proteome, metabolomics, and also bioinformatics. So here, basically, this for omics is our core business. So we have a dedicated fellows and also researcher who are very who are expert in this aspect of omics. So we have CODA for genomic and transcriptomics. And for metabolomics, of course, we offer a comprehensive analysis starting from sample preparations. We do consultation on the sample preparations, and also we accept crude extract, so that whereby we can actually do the sample analysis and also data analysis. So basically, this is the representative schematic representative workflow for CODA metabolomics. So we have the data acquisition. So right now, we are grateful to have our own machine. So we have our LCMS, 𝐿𝐶Q-MS. We also have GCMS, and of course, we have HPLC. Also, we have FTIR to do metabolic fingerprint. And from there, we help you on the data analysis. So we can generate the data, and then we can actually analyze it for you and come up with the interpretations. So for further information on CODA metabolomics, you can actually contact either Encik Syamil Afiq, also Puan Sarah Ibrahim. Or if you want to actually do another analysis of omics, you can always go back to our website, INBIOSIS/EN/CODA to get further information about this service. So with that, I end my talk. Thank you.
Thank you very much, Dr. Kamal Aslan, for your very insightful presentation. And of course, I believe the audience also getting the same thing that I have here. So before we wait for the audience and participants to type the questions, so anyone who has the question, please type in the chat box and I will read for you. But I also have few questions while waiting for the audience to ask. The first one, because you, Dr. Azlan, you have divided into, one is plant metabolomics and another one is microbial metabolomics. I just have one question regarding the microbial sample, right? Because some, if let's say if you're working with fungus, sometimes we grow it in the solid phase. Solid state fermentation, right? So my question is, do we still need to do the quenching?
So we need to still to do what?
Do we need to have quenching for the sample preparation?
For fungus, actually, you not necessarily you may not need to do quenching because all you need to do is you can do a filtrations. So get the cell pellets. I mean, because it's suspension, it's a suspension culture for fungus. All you can do is actually do a filtration. So get this biomass, and from there you can actually straight away go for liquid extraction.
All right, thank you. We have one question from Dr. Tan Janti. Okay. Great sharing, Dr. Kamalrul. Thank you. His question is, in extracellular microbial metabolomics, how do you overcome the challenges in this discerning extracellular metabolites from medium-derived metabolites, especially using nutrient-rich brom?
A very good question, because this is actually a real problem that I faced during my PhD. So basically for the Lactobacillus, we use M70. Is actually quite it's a very what you call it, it's a very nutritious medium. So in this case, what we did was, of course, we also try to employ different media, very basic media. However, that one didn't work very efficiently because we have a very low yield. So basically, bacteria didn't actually grow. So what we did was, we after quenching, we try to wash our cell pellet as much as possible. Try to actually basically remove all these mediums derived metabolites, medium-derived metabolite from the cell pellets, and then only we do the intracellular. Also, we did a control using the non-rich medium. But again, the non-rich medium is quite tricky because not all microbes can actually grow in it. So yes, that is one of the difficulty that we have. The best way is actually, of course, try to cultivate in a non-rich media, and then from there you can do some comparison.
Okay, another question. So because you're handling with many biological samples, so your advice is from your experience, how many replicates that we can say it's safe or significant in giving result for the metabolomics?
Right. Okay. So from a statistician perspective, so the best number for your size sample is 25 and above, or basically 30 and above. So from there, you can actually assume that your data is normally distributed. From there, you can use univariate analysis. But in case if you have a small size sample, for example, due to limitation on the products, on the samples, yes, you can you can actually reduce your size sample. Maybe because of the sample itself is very difficult to obtain, or maybe because of the budget constraints. I think from there, still you need to have the right size sample. So in that case, if you have very small size samples, you need to do normality tests to see whether the data that you have is it normally distributed, and from there you need to choose whether to do non-parametric or parametric. That is from the statistician perspective. In case of multivariate analysis, without this univariate analysis, so usually the safe numbers, if for you to have 5 biological rep with 5 technical reps. So total is 25. Because from there, you can actually deduct your sample if you have outliers. So of course, you need to have at least 3 biological rep for each of the condition with 3 technical rep for each of the biological rep for you to have a good quality data and also result when you do multivariate, especially when you do PCA or PSDA and PSDA. Okay, I think 9 is the minimum, but if you can go higher, for example, like my case for soil metabolomics study, soil, So we have problems because the variability issue of the soil itself. So what we did was we increased the size of the sample to up to 99 sample, just to actually address the variability issue. Okay. But again, it's also depend on the sample, the type of sample you have. For microbial metabolomics, maybe you can have less than what you have in plant metabolomics. But then again, you have to look on the statistician perspective and also the metabolomics perspective.
Understand. Thank you. So we have questions from Tanisha. So highly interesting presentation, Dr. Kamalrul. Thank you. So volatile metabolites are also present in soils. How the untargeted metabolite approach was able to capture the entire soil metabolome, taking into consideration the occurrence of rich and very lowly concentrated metabolites.
Okay, so I think I have to mention that here for soil metabolomics, we use LCMS, so we actually didn't focus on the volatile compound. And then of course, like I say, so for soil metabolomics we have issue due to the low metabolite compound that we obtained. So what we did was we did a SPE, so solid phase extractions, whereby we use this method to concentrate the sample, the extract that we obtain from this soil, and it actually help you to get more metabolites.
All right.
So, but again, we didn't capture the entire soil metabolome. That is until now, no one can actually done that. No one actually have done that because you cannot use only one analytical tools to capture the whole metabolite or the whole metabolome. Okay.
I agree with that because I still think that we struggle to find as for example the extraction that can extract the whole metabolite,
Yes.
whole metabolomes. So that is why either we need to have various of solvent extractions or together with various of analytical methods just to complement the variation of the metabolites that we can capture for the metabolomics approach. Actually, I think it also depends on your experimental design, also what you would like to do. Like, for example, when you look on the soil metabolomics, for example, you exclude the volatile. You don't want to look on that metabolites, right? And so that is why you use LCMS.
Yes.
We have one question from Dr. Imelda. Thank you, Dr. Kamalrul, for interesting studies. Do you mind explaining more on how you do your univariate analysis? How to visualize these data?
Okay, thank you for your question. So for univariate analysis, so for this univariate analysis, what we did was first, after we obtained this the data matrix that consists the metabolites and also the numerical data for each of the metabolites. So first, we did the normality test to see whether the data is distributed or not. And from there, once we ensure that the data is normally distributed, of course, we subjected this data to normalization using internet standard. Of course, we employ internet standard. That is a basic step for metabolomic studies. And then we also did a log transformation, and we also did a scaling method. That one is for metabolic analysis. And from there, we did the normality test using divines or other appropriate statistical analysis. And from there, we come up either we're going to use parametric or non-parametric. So from there, if you use parametric analysis, then of course we can use either t-test or ANOVA to calculate our significance. And as for visualizing this data, I think if you look at the metabolomics paper, most of them, of course, they just most of them use bar graph or bar chart or to actually visualize your data. Also, they use a lot of heat map to actually see how these metabolites are changes according to class or according to group. Yeah.
Thank you, Dr. Kamalrul. I hope you answering the question. So we have another question from Dr. Tan Janki again. He's very interested in your study in terms of the microbial metabolomics. So his question is again, in microbial metabolomics, if we have limited budget, which MS platform, whether liquid chromatography or gas chromatography, that you will recommend to start for untargeted comparative study?
Okay. It actually depend on your objective. In my case, I'm using lactobacillus lattice. So I know that this lactobacillus lattice can produce volatile compounds. And in that study also, I'm focusing on the central carbon metabolism, which I want to look into the amino acid profile. So based on this information, I decided to use DCMS. So because I want to capture the amino acids and also the volatile compound that actually produced by this lactobacillus lattice. So again, it depends on your objective. So if you are looking for a secondary metabolites, for example, like compound that have antimicrobial properties or antifungals or other activities related to pharmaceuticals, of course, you may want to use LCMS because most of these compounds may be indicated as secondary metabolites, which are not volatile compounds. So it's much more easier to be detected using LCMS compared to this. So again, it's based on your objective or what actually you want to extract from your sample.
So again, I think it's based on your answer, it depends on your objective. If I think Dr. Tan Janki, if you would like to do the whole untargeted, so you cannot choose. You
Yes. Yes.
All right. So we have another question from 𝐼𝐞𝐿, our PhD student. So great presentation, Dr. Azlan. Thank you. Could you share more on how you relate the phytotoxicity and metabolomics study? Is there any targeted metabolites that you are interested?
Right. For the Vigilia Turbiata studies, so the idea was to actually obtain what are the metabolites that actually contribute to this phytotoxicity activity. So in this case, we try inhibition on the germination seed, inhibition on the shoot and the root length of the seed that we use as our target. So we actually use so-called Chemometrics. We integrate metabolomics data and also biological data activity. In this case, the phytotoxicity activity of the inhibition percentage of this germinations, again inhibitions, again roots and shoots. So you can actually do that using PLS. From there, you have your regressions, and then you test with because what I did was untargeted metabolomics. So I have a list of metabolites. So I just subjected all this metabolomics data, combine it together with the phytotoxicity data, and then I did the SDA and come up with the metabolite that actually contribute to this phytotoxicity.
Thank you. Answer. So we have another question from Alina. Interesting topic. If you don't mind, Doctor, which faculty or department that you are collaborate with at UNIZA?
Okay. Currently, I'm collaborating with Dr. Nasuha from campus Padang, I think. It's under School of Bioscience, if I'm not mistaken. Yeah.
In Besut, is it?
Sorry?
Is it in Besut? Campus Besut?
Campus Besut. Gombadak, if I'm not mistaken. Gombadak.
Oh, Gombadak. All right. Okay. Question also, Dr. Azlan. So when you do, for example, LCMS metabolomics, right? So because the data, I mean, the metabolites that we get is basically is a putative.
Yes, putative.
So how you verify that is actually the compound, or how do you have any plan? Because the issue is now when I try to submit papers, right? So when I just use MS data, normally they will ask if I have MS data just to confirm it, or so how about you? How you deal with this?
Okay. So of course, nowadays for metabolomics analysis,
Mhm.
they will require us to actually provide MS data to back up the putative identification. So in my case, yes, I provide them with that the informations because but still when I wrote it, I will put it as a putative identification. Although I have the fermentation, although I have the MS analysis, because yeah, of course, because of the budget constraint, I don't have the power to actually purchase the standard or the pure standard of for each of these
Yes. Mhm.
compound. And luckily, until now, they actually accept it. I guess it depends also on the editor and also the reviewer.
Do you have any plan, I mean, in the future to explore on, for example, molecular networking, or like serious
Uh,
in silico tools, for example? This.
yes, nowadays many more and more publication actually
Mhm.
use the molecular network to confirm the
Yes.
identifications of metabolite, whereby they combine the MS data together with MS data. So actually I'm looking into it, but I'm
Okay.
still learning about it, and perhaps maybe on the next paper I will actually incorporate that tools for publication. Yes, it's actually interesting way of you to actually identify and of course to confirm the identification of the metabolite.
Yes.
Mm.
Mm-mm. Agree with you with.
I think
So
perhaps maybe uh, you guys can actually invite somebody who are actually working on molecular network, especially on the identification metabolite for the upcoming webinar. That would be an interesting topic actually.
yes, I think so. I agree with you with that. So again, out of all your presentation, so normally what actually a software that you use for visualization of your multivariate data? Uh, do you
Okay,
only use
so
Synca, or you use various types?
for my study, I use heavily depend on metabolic analysis and also Syncapi. So we are lucky to have Syncapi software. So thank you to Associate Professor Dr. Sharon Dakain for providing us the software. Also, I'm using metabolic analysis because it's a great web-based tool that allow us to actually do analysis of our data and also visualize our data. Other than that, I also actually use iScript to do my visualizations. But that one is for me is a bit difficult because I'm not a frequent user. But there are some website. There are several websites that can help us. Actually, help me in creating our own command for iScript to visualize our data.
Thank you, Dr. Kamal Aslan. I think there is no more question. So again, I would like to thank you to Dr. Kamal Aslan for your time, for sharing us your, I think, very interesting, diverse topic from the plant until the microbial metabolomics. And uh, so we have come to end to the webinar again. Thank you very much, Dr. Kamal Aslan, for your time. And I'm sure
Thank you.
based on your presentation has opened up our view and give an idea for us how we would like to bring our research for in the next level. So basically today's webinar has recorded 58 participants in total, where that we have like 41 from the WebEx and 17 actually from the FB Live page in INBIOSIS UKM. I also would like to thank to all participants for spending your time with us today. So before we say goodbye, as normal, tradition, let us immortalize this session by switching on our video and have our faces captured. So technical team, please handle this session, yeah?
Assalamualaikum and good evening. So, can you please switch on your camera? Sleeping to air. Okay. Okay. So, 3, 2, 1, smile. Okay. Okay. So next one. Okay. 3, 2, 1. Okay, that's all. Thank you.
Thank you to the technical team. All right, so Dr. Kamalrul, I would like to invite you to another breakout session after this. So
Okay.
please wait for the invitation, yeah, to the breakout session. So to the audience, we have posted attending links for today's webinar in the chat box. So please be informed that this link will be available for 30 minutes only. So please click on the link and fill in